are back, and today we are talking about protein folding. And so proteins get their function, like all things do, from their shape. And their shape is determined by their folding patterns. And so there are 4 levels of folding: primary, secondary, tertiary, and quaternary. But before we start talking about the actual folding, we need to revisit the structure of the monomer of protein, which is amino acids. For without understanding the structure of amino acids, the whole story of protein folding does not make any sense. So if we turn our attention to the left side of this whiteboard here, we have a generic amino acid structure. Now this is not any specific amino acid because it does not have a defined functional group. Okay. So all amino acids have 3 main parts. They have the amine part, they have the functional group, and they have a carboxyl group here. Now, the amine and the carboxyl group are just used to link to the next amino acid. Right? So if you imagine train cars, right, this is like the front of the train car, it links to the back of the next train car. This back of this train car would link to the front of the next train car, which would be the amine group of the next one, would be the carboxyl group of the previous one. Right? So the only chemically active site on an amino acid is this functional group here. Okay? And so whatever chemical properties the functional group has, those are the properties of this amino acid. So if you put an acidic functional group on this base, then you have yourself an acidic amino acid. If you put a hydrophobic amino acid in this spot, then you have yourself a hydrophobic functional group in this spot, then you have yourself a hydrophobic amino acid. Right? So whatever group you put in this R slot here is going to determine what properties this amino acid possesses, which is going to become very important when we look at how the protein gets its shape and its functions. So the primary structure of the protein is really straightforward. It is quite literally just the sequence in which the amino acids of this protein show up. And so that sequence was determined by the coding in the mRNA and the codons in the order in which they showed up, which was in turn determined by the sequence of nucleotides in DNA. Right? And so ultimately what this is coming down to is the coding in DNA determining what amino acids are going to show up in what order. The order is very important because the order determines the way in which they will bond with one another and create the folding steps that we see coming up. So secondary folding comes into play once you already have your amino acids lined up. Okay. It begins as soon as the polypeptide starts coming out of the ribosome. Right? Once you have a reasonable length of strand, these bonds start forming, these connections start forming. Okay. And the secondary folding is driven almost exclusively by hydrogen bonding within the protein between different amino acids. And so here I have 2 examples using 10 amino acid long proteins. Now, proteins are, hundreds of thousands of amino acids long, but here we are using 10 because I only want to draw 10. So that's what you get, right? And so each of these has the same amount of amino acids, right? But by altering the order in which the amino acids show up, we change the way in which the amino acids will bond with one another. So in this first model, I have a red amino acid and I have a blue amino acid. The functional groups of the red and blue amino acid make it so they want to hydrogen bond with one another. Okay? In this first model, I put them in these positions. Right? And so when they bend up, they'll take on this shape. Okay? So same amino acids, just in different places. Okay? Takes on this shape, as opposed to if I put them in these positions, this protein might take on this shape. Okay? And so these hydrogen bonds are the secondary folding. And it's not going to be this simple. There's a lot of different hydrogen bonds happening at a lot of different times. Right? But that's the general gist of it. So different areas of hydrogen bonding result in different bending patterns. Once the secondary folds have completed, we start to see the emergence of the tertiary folding. And so the protein already has its general shape. It looks pretty much like the way it's going to look when we get to this step. But there are some slight tweaks that can occur here. And so the major way in which these tweaks occur are through hydrophobic and hydrophilic interactions with the aqueous environment. Right? So if we look at this model, these two amino acids here, labeled with blue, have functional groups that are hydrophobic. Right? So the hydrophobic molecules want to stay away from the aqueous environment. So this protein, even though it has its general shape already, will bend inwards and encapsulate those hydrophobic regions to protect them from the aqueous environment. So you see this little invagination here caused by that bending. Right? And so that kind of thing can happen in tertiary folding. And the other thing that happens at this level is the formation of supporting bonds. And so the general structure is already laid out, but there's still some different amino acid groups that might like each other, and the functional groups might form bonds with another. So these are very often covalent bonds. So these bonds are nice and strong, right? And they help the protein keep its shape. And one of the most common form of bond that we see here is the disulfide bridge. And these are formed when two functional groups that can interact with one another both have a sulfur and a hydrogen group on them. Right? So these sulfhydride groups will form these disulfide bridges that help the protein keep its shape pretty solidly. So once you have your tertiary folding complete, this protein shape is nice, it's solid, it is what it is, it's happy, it's ready to go do its job. Okay. There is a fourth level. Not all proteins require this fourth level. Okay. This quaternary level is when you have more than one protein engaging with one another. And so they form these big protein groups, these protein complexes. And that combination of multiple different proteins together to form one big protein is that quaternary bonding. Okay. So those are the 4 levels of folding, right? Pretty straightforward, pretty simple, not too bad. Just as a general overview, the folding gives the proteins their unique shapes. And of course, as you know, the unique shapes determine the unique functions because all shapes are determined. Nope. All functions are determined by shape, not the other way around, Mr. Lavier-Yagoon. Okay. The folding of the protein will leave some functional groups unbonded and exposed to the environment. The regions of the proteins that have these exposed functional groups will possess the chemical properties of those functional groups. Right? So a lot of the functional groups are already tied up in different bonds, or they're tucked away inside the protein, and those guys aren't playing any role in this. But the ones that are sticking out will control the chemical properties of the protein. Right? And so like when you're looking at things like enzymes and stuff, these sticky out parts are the parts that are actually engaging with the world and doing the work. So the functional groups are extremely important because they provide the function. It's almost like somebody named them really well. Right? And the last little point is that different areas of the protein, different regions of the proteins will have specific jobs for the overarching larger job of the protein. Right? And so if this protein's job is putting Jell-O into a Jell-O cup and then putting a spoon into the Jell-O cup and then serving it to you at the restaurant, right? There's a domain of the protein whose job it is to put the Jell-O in the cup. There's a domain of the protein whose job it is to put the spoon on top of the Jell-O, and there's a domain of the proteins whose job it is to bring the Jell-O out. Right? And so that whole thing, its job is to deliver Jell-O to you at the restaurant, right? But each domain has its own little role in that part. Yeah, that's it. Protein folding, done. Have yourselves fantastic days. Bye.